This is drawing 45, leukocytes part 8, how T cells function. And this is going to have a lot of cartoons on it. You need to have this printed page so you know, have those lines in here, so know kind of where we're going here. We'll start with this top part. I'll zoom in so we can hopefully read this. Alrighty. Okay, so it says here, the macrophage, or a dendritic cell, but we're going to draw a macrophage here, presents a piece of pathogen to a naive T helper cell. At the same time, the macrophage will secrete cytokine messages that will enter the T cell and tell it how to differentiate. So right here, we're going to need a macrophage. Now, remember this guy, I'm going to make these cartoony ones. I can make one of his arms kind of go down here. Around here, going to give him a little arm. Remember, these guys can change shape. They're like an amoeba. They can kind of be any shape. So whatever shape you draw is good. So except for maybe make sure he's got an arm over here because he's going to meet a T cell over here. I'm going to give him eyes, and I'm going to make a more cartoon version. Okay, and I'm going to give him a big mouth to make sure we remember that he is a macrophage, big eater. He's our big eater. Just for fun, I'm going to give him a CD31 hook. Remember, he's got a obviously not this big. This is way, way out of proportion. It would be microscopic on this, just a little dot here. But the CD31 hook is the thing that it goes and it polices the neutrophils. The neutrophils also have a CD31 hook, and it hooks together like that, and the neutrophil, if it's sick, it won't be able to get away, and then it'll get eaten. So this is this policeman's hook for the neutrophils. And I'm going to give him a bunch of little things like this. Just remember that it's got receptors. Very important to remember that the macrophage has various kinds of all kinds of receptors. We're not going to go into all the different kinds because that's way too much information. But it has ways that if these touch another cell, a bacteria, some of a pathogen, that it'll trigger certain things to happen. So it has that's like its eyes, right? It doesn't really have eyes, but these are like the feelers where it can feel different things, and each receptor can only feel one thing. It's got a lot of different ones. And one of the most important receptors here it's got is this little MHC clip, and we can even focus in a little bit more here. We'll zoom out in a minute. So we got, this is an MHC2, and I want to draw one right over here. So remember, this is MHC2. And only the antigen presenting cells like this guy have number 2. All body cells have MHC1, and that's how they show what's going on inside. But these guys, remember, have MHC2. And that is handy because, if you want to make some lysosomes, you can give them, yay, lots of lysosomes for digesting pathogens. Okay, so this MHC2, I'm going to put a little dot here. So it's got a little piece of pathogen. It's got an antigen. Right, we call it an antigen. Right here, it's presenting this antigen, holding it out to show it to hopefully some T cells that are wandering by. And here it is. I'm going to draw, when I draw the T cell here, I'm going to draw his little receptor first, and then I'm going to draw his body. He looks like the letter T. Right, so it kind of looks like he's got a T-shirt on, sort of big head like this. That's his cell part, right? Eyes that are kind of looking up, going, ooh, little mouth. I don't know what to do. Gee, I don't know anything. He's naive, and I'm going to put like across his shirt. You know how they have like names across the back of football jerseys? Well, I'm going to put helper across his. And just to remember, CD4, the helpers. And he's got number 0. His jersey says 0 because he is naive. Doesn't know anything. He doesn't know what to do yet. Okay, so here he comes. But it just so happens that the shape, remember these guys, the helper T's, And well, Altis, they have this receptor that is very, very much like those Y-shaped antibodies in that they have a special shape at the end, a unique shape. So a million helper T's can come by and run into this, and they'll just go, I don't know what you're holding. It doesn't mean anything to me. But if the right one comes along and he feels this, and he'll go, ooh, that matches. This is important to me. And he'll stick to it. He'll stick around, okay? So if he locks in on it and says, you know what, that's a match for my receptor. I think I'm the right person for this. What happens next is that then signals that it causes the macrophage to release some cytokines. So I make a whole bunch down here and then less. It kind of looks like they're coming out. If you make it look like they're more dense down here and then spreading out like this, it looks like it's kind of direction like that. Okay, so these are cytokine or cytokine messages. That's how cells talk. They send chemicals back and forth. So this locks in that the macrophage responds by releasing these cytokine messages, little chemicals that go across, and they're going to tell this helper T what to do about it. He's go, okay, I'm ready to do something, but give me a clue what. Well, meanwhile, the macrophage has interacted with the pathogen, right? He ate it, ground it up, put the pieces in the lysosomes, and we saw how that happened. So he's had some reaction, some interaction with it, and with the help of some of these other receptors, there's a series of events that cause certain cytokines to be released. Okay, so if it releases a certain type, it's going to cause the T cell to do something. If it releases a different type, the T cell will do something else. So it depends on the chemical message coming from the macrophage what this helper will do. Okay, we can put tells helper T what to do. Okay, and that's what it says right here, that these cytokine messages will tell the T cell how to differentiate. Okay, so the main thing it needs to know is whether to look for the pathogen inside the cell or outside the cell. Remember, we looked at some pathogens pretty much stay outside the cells, and others love to invade and live inside the cells if they can. And that's very important to your immune system to know how to fight, inside or outside. So we're going to give it these two options here. One option over here, if you draw kind of a long line stretching over to here, that's kind of long, but we're going to go, this is one option that it could take, and this is going to say, look inside the cells. Look inside cells. And the option that's going to go down here, this option is going to say, let's see if we can see, sorry, I'll move the paper up here, okay? Look outside. So this is look inside the cells and look outside. Now, if you look inside the cells, this is a very important word here. I'm going to make it just, it's going to be like a side note to this drawing. I'm not going to make a big deal about this vocabulary word. However, when you are reading a regular textbook, they're going to make a big deal out of it. Okay, so I'm even going to put it in parentheses, okay? It's called cell-mediated response. Okay, and this just basically means cell-to-cell warfare. This means you're going to go and you're going to attack. You're going to do hand-to-hand combat, cell-to-cell combat. Okay, cell-mediated means cell-to-cell combat. Looking outside, this is going to be called the humoral, H-U-M-O-R-A-L, humoral response. Now this is a strange word, the humoral response. What is a humoral? Well, you see a little note here. You can put a little asterisk there, these little things that goes chick, 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 chick. Put it right there, and then come down here for this note down here. See this note? Okay, we'll put a little asterisk there, and you can put the same little asterisk down here, and that means when you get to here and you see this little asterisk, you can even make it pink or yellow or something and color code it. That means come down here and look. Humor is a word from the Middle Ages. It means body fluid. Therefore, a humoral response is one in the body fluids. And if you know anything about history, they had this theory of body fluids that you had the 4 fluids. You had blood was one of your fluids. Phlegm was a fluid because, you know, it's the stuff you cough up. They're like, hey, it's a body fluid. Bile, I think they had like black and yellow bile, 2 kinds of bile were your body fluids. And they thought mistakenly, but they thought that your body had to have a balance of these fluids, and when you got sick, that was caused by an imbalance in the fluids. So they would do these crazy tests that are, I mean, of course, ridiculous because they were wrong, but they would try to determine if you had too much of something. Well, what would happen if you had too much blood? Yeah, they drained some out, and that's what happened to George Washington, first president of the United States. He was sick. They determined that he had too much blood, so they drained out like half his blood and killed him. So just don't forget, doctors killed George Washington. So they don't do bloodletting anymore, fortunately, because they had no idea really what blood was. They didn't know how important it was. So inside the blood, poor George Washington, what was happening in the blood? His body was fighting. His body was trying. It was doing, probably, if he had an infection, his body was doing a humoral response, and they drained it off. So what was happening in the humoral is your body is going to fight in the fluids. It's not going to do cell-to-cell combat. Your body is going to dump those antibodies, those Igs, into the blood, which is your body fluid. And so you're going to flood your body, your blood, with all those little Y-shaped antibodies, and they're going to go, and the antibodies are going to go and grab onto the pathogens. Okay, so that's the difference here. And I say books make a big deal of it. Cell-mediated is cell-to-cell combat, as we're going to see. This is like, I'm talking cell-to-cell combat. Yeah, like the T cells have little, like, basically guns that they shoot the infected cells, okay? Hand-to-hand combat here between the cells. And the humoral response is going to be, go get those B cells and tell them to crank out those little Y-shaped antibodies and dump a huge bunch of them into the blood and let those little antibodies go around and find the pathogens. Okay, so we'll take a detailed look at those. However, before I go to take a detailed look, I want to mention, see this blank space here? I want to mention there's a couple other responses it could have. These are the major ones, and usually they don't even really mention this, but I want to mention it so that you've heard of this. Okay, there's other things that can happen with T cells. There are other types. We're going to talk about T1 and T2, the helper 1 and helper 2, but there's a few other ones, and I just want to make sure you've heard of them. Over here, let's see, we can finish this guy's little hand here, okay. We're going to do 3 other little arrows here. One here, and one here. Okay, so there are a couple other kinds of T cells that you need to know about. All right, draw a little tiny one here. We're going to make these small so they don't take up a lot of room. Circle, give them a little T shirt, ha ha ha. Give him a couple little receptor arms. And on his jersey, you're going to put R-E-G. Now, he's a helper. I mean, that's kind. I'm going to write helper on him. Reg stands for regulatory. Regulatory. Regulatory T cells tell killer T's to stop. Okay, so I'm going to put a little stop sign in his hand. He's like the little traffic guard. See, 1, 2, 3, 4, 5, 6, 7, 8. Try to make a little 8-sided thing there. I'm going to write stop on it. He's holding a little stop sign. You can make it red if you want. Okay, he knows what to do. He's got a smile on his face. He's happy. He knows what to do now. Okay, see him there? So he could turn into a regulatory. Now they used to call these suppressor. To suppress something is to lessen it, to dampen it down, to make it less. They used to call them suppressors. So if you see T suppressor cells, that's the same thing. But regulatory is now the preferred word. So we're going to go with the current word, and that's regulatory. So the regulatory cells will tell the T cells, stop, it's enough, infection is over, calm down, quit killing things, calm down. And in people that have autoimmune responses to infections, and this is a lot more common than I think even doctors realize, and someone in my family has this, so I know this, that they have an autoimmune response to infection. Infection starts ramping up, and the T cells get way too excited, and apparently there aren't enough regulatory cells. And there's been experiments that suggest if you can take some of these out of somebody, get some of the regulatory cells, and then multiply them in a test tube, get a whole bunch of them, and shoot them back in, calms it right down. But it's really difficult to do. It's not really something that your standard doctor's office does. So it's a little bit difficult. But anyways, these regulatory guys are really, really important. There's not a lot of them. There's only a few produced, but they are extremely important. Just want you to know about them, that there are these T cells out there that tell your rest of your T cells, okay, enough, calm down. Very important, okay? Good to know. Okay, so down here, another thing that could happen is we also have another kind of cell. He's got a different number on his jersey. Let's make him right here. Okay, he's got number 17 on his jersey. And he's all happy because he knows what to do. So, but he's going to be holding an arrow pointing up. Okay, so unfortunately, these guys, what they do is increase inflammation, 2 M's. They increase inflammation. So this is kind of not really a smiley face for your body sometimes. And remember, inflammation is important. It's necessary. Inflammation is part of the body's defense system. However, you can get too much, and then it starts damaging. And these little T17 cells, if there's too many of them, they get out of balance. They can cause too much inflammation. And sometimes, the same thing, they can start attacking body cells and things. So these guys they're necessary, but they can also be problematic if they get out of hand. Anyways, so there's these T17s. You just need to know about it. They're there. And the last kind, there's more, but I'm just cutting it to 3 here. But it's really good to know how complicated the body is. It's very important to know that it's not a simple thing. And it's important to know that we don't know everything, and we're still finding out things. Okay, so this guy, once again, he's got a smiley because he has a job now. And he is number 3. And so I'm going to put helper across his chest. I should have put helper on the last guy too. You can put helper. That's his name. Okay, and so number 3, he tells B cells to make IgAs, which will clear pathogens stuck in mucus. Okay, so it's going to tell the B cells. Now the B cells, remember, on the little Y thing. Okay, the antigen, the antibodies look like little Y's, right? And this part, this is where the shape matches. This is where right here on the T cell, it looks like that. It can handle 2 of them. This part here, these parts are where the nasty foreign thing sticks. And this part, the base of the Y here, right, this is the one that sticks to your body cells. This is the one that can be A or G or M or E. There's different types of sticks. E is the stick that causes allergies. M is the one that the B cells make first when you first have an infection. G is the one we'll see in a minute G is the one that really fights infections well. And IgAs are basically for clearing stuff out. They're found a lot, like in your nose and your lungs and your intestines. And they just help you get clear. Remember, it's the mix-up between E with the really severe algae problems. Sometimes mistakenly, your body puts an E stick on it, and you have these terrible reactions to things. A is just supposed to help you clear out. You're just, it's very non-inflammatory. It's very innocuous. It doesn't make you sneeze and all that. These A's aren't much of a problem. So this guy is actually kind of good. It tells B cells to make IgAs. This is a very calm. This is a very if you're IgAs, I think maybe these guys are the ones that are involved. I said in another video, I used to be allergic to cats. My body was cranking out IgEs to cats all the time, and now I'm not allergic anymore. I think maybe I don't know, was this guy? It's just speculating, but I think now my body just makes IgAs to cat dander, which is what it's supposed to do, and I get rid of the dander and I don't have a big allergy attack. Anyways, just wanted to mention there are also T3s. And they have this role. They help your body make IgAs. Okay, so, all right, let's look at these two reactions here. Let's look at this one first. I'm going to go down here. I'm going to zoom in on this side. Actually, I'm going to zoom in right here on the top so you can see. And we are going to make a helper 1 right here. Let's put his head right about here. Nice big head. And of course now he was naive. He didn't know what to do. He didn't know whether he'd ever be needed or not. But now he's got a big smiley face because he has a job. He knows what to do. So he's no longer 0 on his jersey. We're going to call him number 1. Okay, and he's going to be helper. And if you want to reinforce it, I keep writing CD4 so you get used to that because you'll see CD4 a lot. Helper number 1. So what does he do? He is going to, number 1, we're going to slide this down a little bit. See here, it says the TH1, so now we have him right here. The T helper 1 will make cytokine messages that will stimulate the macrophage to speed up. So we need to make little cytokine. He releases chemicals. Remember, this is all automatic. Your body, it's like the little machine. These guys, the cells can't really think. They don't have brains. It's just automatically the shapes of the molecules, they just trigger it. It's like a very cleverly built machine. Okay, so these are cytokines, which are messages. And just in case you're interested here, this brand, these are called interferons. Okay, interferons are famous for interfering with viruses. They help your body to interfere with the viral replication to fight them. Anyway, so that's what this kind is. There's lots of different kinds of cytokines. There's interleukins and all kinds. These happen to be interferons, just in case you're interested. So the messages go over and it tells the macrophage, so here's our picture, it tells the macrophage to speed up. Go, eat more, eat, digest, process, eat, digest, gobble, gobble. It tells it to go into overdrive and really, really get killing. Just kill those pathogens left and right because they're something to be concerned about. Okay, we really want to. This could be a threat to the body. So it stimulates the macrophage. Funny thing is, it also stimulates itself. There are some chemicals where if you can make a loop, he's going to be releasing these chemicals, and the target receptor, like for these chemicals, have to have receptors. So the macrophage up here is going to have receptors that fit little mailboxes that the mail can come to. And the funny thing is that sometimes they release a message for themselves, and it circles around and it goes into its mailbox down here. So that's what's going on down here. The T helper 1 also sends messages to itself telling it to start making clones, it's telling itself to get upset about this pathogen. Okay, it's like having a conversation. Self, should we get upset about this? Yes, self, I think we should. Okay, all right, so it's going to start making copies of itself. Right, it will be clones. It'll go through mitosis and just start making copies and copies and copies. So although we're doing, we're just seeing one of these, it looks very simple. We're just going to draw one little guy, right? It's really, in actuality, it's an army of T's that's going to be produced. So what is it going to do now? Well, okay, T helper 1 has the job, we're going to make it right about here. This little head. He has a job of talking to other T cells. So let's make him have nice bright eyes, and then in fact, his eyes could even kind of be looking over this direction. He's opened his mouth, he's talking, we'll give him his little receptors. Okay, and then over here we need to have a T cell and then an edge of a body cell. Maybe I'll just put the edge of the body cell in already like this. So we got that in there, okay? And then the body cell is going to have an MHC1 clip. And the clip is holding something. And in this case, it's probably going to be something nasty because remember, in this body cell, the MHC1 clip, my house cleaning, the body is cleaning up and showing all the time, showing what's going on inside the cell. So unfortunately, I'm going to write the word infected here. This poor cell is infected with a virus. So what kind of nasty things, pieces of the virus might show up on here. And so this T cell, put a little receptor here. I'm going to draw the receptor first. There. And then we're going to draw the T cell onto it. Now don't make these guys touch. They're just standing there. This guy is standing over here. Okay, and he's wondering in a bubble, here's this little thought bubble. He's wondering, should I kill this cell? So he's pretty sure, he's latched onto it. Remember, they don't have brains, but the shape matched. So 1,000,001 T cells can come past here. They don't match, but he matched this little pathogen shape. Now remember, he's trained. So this one, he's a TC, and I'm going to write killer on his jersey. You can call them killer T's, but remember, in books you'll often see nowadays they prefer to call them cytotoxic, and that means he's got a gun. They're CD8. In fact, you can go CD8 killers. And he's got a perforin gun. He hasn't used it yet, but he has the capability of killing. These helper guys, they don't have weapons, do they? Nope, no guns on him. Just guns on the CD8s, on these killers. Okay, so he's sitting here. He's matched. He's shape matched. And he's latched onto it. His little receptor. He was trained to recognize antigens that are not self. So he came across something. Had this infected body cell been presenting, I don't know, a piece of its own ribosome or a piece of something from itself, this guy wouldn't have cared. But this, he was trained to recognize in the thymus, right? Trained to recognize pathogens. So he's latched on here. However, we want to make really, really sure that this is really okay to start killing, because this is a body cell. Before we start killing body cells, we have a double check system, okay? So it's thinking, should I kill this cell? And so the job of the T1 helper over here, he's going to say, just put the word kill. He's going to give him the okay. And how do they communicate? Yep, he's got to send little chemical messages, cytokines. Okay, so these are cytokines. Cytokine means any chemical message that goes across. And I think in this one, it's maybe it's called interleukin 2. The white cells, when the white cells talk to each other, they have a special code called the interleukin. Inter, between, leuco, white. The interleukin. He uses their code. He uses interleukin code number 2, which says go. So if he releases that, then bang, this guy, let's make sure he's number 1. So he needs helper number 1 to confirm, yes, go ahead and kill that cell. So what happens now? Let's look at how they kill. Of course, you all want to know how the gun works, right? Of course we do. We want to know what's in that gun. So let's draw a line here, though. 2 alternatives. Now remember, these guys are multiplying, making copies of themselves. We're almost down to the, I'm going to zoom out just a little bit because I want to make sure to get this on the, so you can see the bottom of the page is right there. Okay. So TH1 activates the TC cell. Right, so I was just saying what we just showed right here. T helper 1 activates this killer cell. TC, C is for cytotoxic. Okay, so one thing that's going to happen is it's going to make lots of killers. It's going to start duplicating itself. And most of the copies are going to become killers, but let's put over here a little guy. With his eyes closed, he's remembering. And he's a killer. T sub C there. Cytotoxic. You can put killer on his chest. However, he's a memory cell. A few memory cells. So it will make, got his receptors there. The T cell will make a lot of killers. However, a few of the killers are going to be inactive. They're going to go to sleep and they're going to remember. So the next time this happens, they're ready to go. So I'll make a few memories, but most of them are going to be killers. So here's where we get to show. We're going to make the body cell here. And it's got, I'm going to draw it this way. I'm going to draw the gun first, then I'm going to draw the guy. So here's the perforin gun shooting a hole. It's putting a hole into the body, into this infected cell. Or it could do this, it could put a hole, whatever, maybe it's into a bacteria or something, but mostly the virus into an infected cell. It could be a tumor cell too, right? If the protein that it's expressing is bad, maybe it's not a virus, but it's a bad protein. Like, you shouldn't be making that. Okay, they can kill rogue cells that are growing wrong. Okay, so it can do that too. So now we're going to put him onto, it's a little hand. We'll put his little arm over here. Now we know where to draw him. His little receptor thing. Of course, his receptor isn't really a hand. This is all very silly, but it certainly helps to remember. Okay, so now he's got eyes looking over this direction. He's got a big smile because he knows what he's doing. But we could kind of give him eyebrows like that to make him look kind of like, he's not mean, but he means business. He's a killer. TC for cytotoxic, killer. Remember, CD8. So this is his, I call it a perforin, P-E-R-F-O-R-I-N, perforin gun. It's not really a gun, but it does shoot little things. Okay, so he's got a little mechanism on his plasma membrane that can shoot out the little bullet things that go towards the cell. And right down here, I want to do, in this last little bit of space here, how does perforin work? I want to make a little line like this. This is the body cell. Okay. And it looks, the bullets come out of the perforin, the little bits of perforin. Let me show you a picture. Okay, it would help. I'm going to zoom out here for a minute. So each little perforin bullet looks like this. There's just one of them, and it goes down and it slides into the membrane. Okay, and then so it stretches all the way through, and then another one comes along and hooks onto that, and another one, and before you know it, you have a ring of these little long-shaped bullety things. They make a ring, and you see the arrow? It makes a little portal, a little hole, so stuff can go in and out through there. It just squeezes the phospholipids away, and the little hole is it looks like a little tube. It really does look like a little tube. It really does. I'll show you. Here's a computer-generated image of it. Isn't that cool? I mean, it really looks like a little tube. Here's what the protein looks like. A little protein map. Okay, and then so that's where the protein, what they just showed you in the map there, that thing, it fits in right there. A whole bunch of them go in a circle to make that. And there's an actual, I'm trying to show you what it looks like. That would be an actual like photograph of a real one with an electron microscope. And then I have one more picture that shows it. You're looking straight down. So these would be the phospholipid heads of the membrane. And these would be the top of the bullets, but you're looking like down through the membrane. And see these little things here going in? They're on their way in. Or if you're looking from the inside, maybe they're coming in at you. But after the perforin bullets go in, make the hole, then the cytotoxic T cell puts these things called granzymes in, these other little toxins, poisons. It doesn't just make a hole. The hole might destroy, you know, the cell, just because you have a hole in it. I mean, that's bad. You don't want to have a hole in your side, right? So the cell might die just from the hole, but the cytotoxic T cell wants to make really sure it's killing. So then after it puts the hole in, it injects these little proteins in. What they do is they go into the nucleus, and there's an enzyme in the nucleus of the cell that is preventing the cell from dying. And it tells that enzyme, stop preventing the cell from dying. This cell needs to be destroyed. And so it activates that enzyme, and the little enzyme in the nucleus just goes and it shreds the DNA. It just chomp. It just starts destroying the DNA. If you don't have DNA, you don't have a living cell. So that's how these things work. We're used to thinking of poisons as kind of a neurotoxin or something. But when you want to destroy a cell, the best way to do it is shred the DNA. So these things start the DNA shredding process. So that is how a T cell can kill a cell. So I just want to put down here a little, zoom back in again, a little like picture of, like if this is the, make it like a little oval, and this will be like the top, make little circles around. So these are like the little perforin bullet things, and then draw some lines down so it looks like a tube, sort of. Okay, and then you could put in an arrow to show, and some little dots. If you want to know what they are, they're called granzymes, G-R-A-N, not like enzymes, but granzymes, toxins. You can just call them toxins if you want, because they're bad, and they go in. So these are all perforins. So that's how, I could draw a little line here. So that's, doesn't have anything to do with this guy. So that's how this perforin bullet, that's how the cytotoxic T cell kills the body cell. It puts these little bullets that make a little hole, and then it sticks a bunch of poisons inside. So now let's go back and look at the humoral response. Okay, so we're going to go up here, back to where we're going to go to look outside. So that's what happens inside the cells. You got to kill the cell. Unfortunately, once a cell is infected, that's it. Cell's gone. Got to kill it. So this side, if the cytokine messages from the macrophage say we need to look for this parasite outside the body cells, then helper 0 turns into not helper 1, but we're going to draw him as helper 2, because we already have helper 3, so we know it's not number 3. Helper, put CD4 if you want to remember, helper number 2. So now it's happy, it has a job, it's helper number 2. Put those little receptor things on there. Okay, helper number 2. What are you going to do? Well, he's going to go talk to the B cells. When you want to find something outside the cells, the best thing to do is to cooperate with B cells, because they're the ones that make all those little Y-shaped antibodies. So he's going to go and talk to a B cell. So we're going to make a T and a B right next to each other here. And the tricky part is they have to be joined, they have to be touching this antigen. So the T cell with his little receptor there touching that, and then make a B. You can also make a over here on the B, what I want you to make is an MHC2. If you can believe it, you know how to draw MHC2. It's that little rectangle with the bite out of it. So you're going to need to draw, so zoom in a little bit here, an MHC2 on the side of the B cell. Here's his head. Okay, so this guy is activated helper 2. This guy still looks a little bit confused. He's sort of doesn't know exactly. He's not maybe totally naive, as we'll see in a minute, but here's his little stick arm there with his little Y. And he's got something here. He's holding in his little, well, it would be in the Y's, it would be more correct. It would be he's holding it if we put it at the ends. Maybe he's caught too like this, okay? So it doesn't look like a receptor. That's a little bit more accurate. And he is thinking in his thought bubble. Give him a really big thought bubble. And he's thinking, I found this thing and made IgMs. And his question is, should I switch and make IgGs? So he found this little piece of pathogen, and of course he processed it. Then what happened is the other cell, this is the little extra tidbit of information about APCs, antigen presenting cells. I think we mentioned it before, but I didn't emphasize it. But there's 3 types of cells that can present antigens: macrophages, dendritic cells, but also B cells. Okay, so here he can take in, this little Y can go in, he can pull the little Y inside and then put the little pieces of pathogen back out on an MHC2. So this is an MHC2 clip. Well, what kind of cell matches with MHC2? The helpers, T helpers, okay? Not the T killers. The T killers can't. Only the helpers, the CD4s are the only ones. So this guy has found this, and then I want to make a little picture of an IgM. They're kind of cool. IgMs, just show you real big, real quick. IgMs have they're those Ys, okay? They're antibodies, but there's 5 of them, all stuck together. It's like dogs or foxes having their tails stuck together or something. I don't know, tied their tails together. They're tied together at the bottom. 5 of them are kind of stuck like that, so that each one can like bind like this. So it's a little bit clunky though. Like if there's a pathogen down here, you know, these guys are just kind of hanging out up here. I don't know if they can bend down to reach. So it's a little bit clumsy. there's a good chance that something will stick. I mean, there's some advantages to having this, but there's also some disadvantages to it. So this guy, this B cell, he said, I'm pretty sure I found this thing, and I was pretty sure it wasn't supposed to be here, so I'm going to make a little 5-sided IgM here. I made some IgMs against it, but I wondered, this is IgM, but should I make some IgGs against it? IgGs are much more effective. Now, the decision is important because once it switches and it starts making the IgGs, it can't ever go back ever again to making IgMs. The information will be gone. It will forever be making IgGs. So we don't want to put up a fight. We don't want to get all upset if there's no need to. So basically, it needs to ask the T cell guy, and he's going to be saying something here. He needs to confirm, the B cell needs to confirm, should I go to war and start cranking out IgGs? And if it matches, of course, in our scenario here, the T cell is the one that has to say yes. How does it do it? You guessed it. Cytokines. Cytokine messages go over. Okay? Cytokines. All right. And so it confirms to the B cell, yes, we really do want you to start making antigens. This is a really nasty thing. So the B cell to become activated needs a T helper 2. Okay, so what's going to happen here is he is going to start making a zillion replicas of himself. And just like we saw over with the T cell, though, a few of those replicas are going to be put to sleep. Right? A few of them are going to be told, and I'm going to put mem on his chest. He's got, whoops, I'm sorry, I made him look like a T. Accident there. Make him look like a B, right? He's B mem, and then we'll put his little, let's get his little arms and everything, and maybe a few extras. He's probably been making a few extra of these too. Okay, so he's got some antibodies hanging off, but he's going to be put to sleep. His job is going to be remember. So the next time around, we're going to wake you up, okay? And you can know how to do it real quickly. Okay, so, but most of the guys are going to go this direction. Lots. And this is just a few. Few this direction, most there. And what's he going to do? Oh, he's going to be the happiest little B cell factory. Make it look like that. He's all happy. And he is producing, or she, antibodies. And this time, they're actually going to come off. So you want to make lots and lots look like they're, and if you make a little V and then put on the stick, if you want to make them fast, I find it's easiest to make the V first and then add the stick. So you want to make many. This B cell will start making, and remember, it'll be all the same shape. It's not going to make lots of different kinds. It'll be all the same kind, but we know it's the right kind because we confirmed with the T cell. Do we really want all these antibodies in the blood? Because you don't want your blood being clogged up with too many antibodies. That can really cause problems if you don't need them. So the T cell had to say, yeah, we want lots of these. So now this becomes, it's called plasma. This will become a plasma cell. So this is I'm going to put a note in the bottom here, short-lived plasma cell. And it is important to remember that they now call it a plasma cell. It changes its name. And this memory, of course, would be long-lived, right? And they can live. These things hopefully will live maybe for your whole life, right? Once you've had the chicken pox or mumps or something when you're young, then you don't have it ever again. So this crank, and these things are IgGs. The stem on this is the G-type stem, which is very important to immunologists, that it's a G-type stem, especially if they're doing blood work. If they find a lot of IgGs in there, that tells them that that's what's going on right here. The B cell is talked to a T cell and has been told to do this. Okay, one little final note. and I want to say one thing that can go wrong with these T helpers. You can see they're very important. The helper cells, that they don't have any weapons, they don't actually do any fighting. They're the ones that tell the T cells to fight, and they're the ones that tell the B cells to make antibodies, the IgGs. So what happens if you don't have these guys? Well, this is what happens in HIV, AIDS. The HIV, the AIDS virus, goes in and the virus attacks helper cells. Can you believe it? It's a pathogen that's designed to attack T helpers. That's not good, is it? Because then without these T helpers, you can't activate these and you can't activate these. So you just get a regular, just say you get a virus, a cold virus, okay? You got the infected cells and the cytotoxic T cells lock on. We need to get rid of these infected T cells, and it goes over and it's waiting for a signal from the T helpers, and the T helpers are just, they can't do it. They're sick themselves. And so they never get the signal, and the cold virus says, ha ha ha, you can't get me, and it keeps infecting more and more and more, and you can get really, really sick just from a cold virus. So anyways, just as a little side note, something about T helpers, the AIDS virus, the HIV, which causes AIDS, is when you actually are sick with it. You can have the HIV, you can have the virus and not be sick. You can just be harboring the virus. But anyways, the virus that causes HIV and AIDS attacks T helpers.